Welcome. Tonight we've also got Hummingbird's drummer and molecular biologist, Dr. Mark Temple.
And Mark has been teaching us how we might be able to use our ears to read code, genetic code, of all things. So over to you, Mark. How do you do that?
So I've got to say that when I hear you introducing me, I'm always surprised that I used to be a musician and I am now a molecular biologist. So if that sounds odd to you, it really sounds odd to me as well. So the question is, I'm a drummer. So I think of musical patterns. I think of repetitions. I think of, all of this repeating sequences, because that's what I do in drums. It's just repeating sequences. So as a molecular biologist, I look at DNA sequences and I see A's and T's and G's and C's, which are the bases in DNA. And there's millions of the bloody things. They just go on and on and on. So it's these repetitive patterns. So I'm thinking repetitive patterns as a drummer, and then I'm seeing sequence. I'm thinking, can I think of that sequence as simple repetitive patterns and start to think about it in terms of beats and in terms of music? So I thought I was going mad when I was thinking about that. I was thinking, no one's going to take me seriously. But then I realized there's an area of science called sonification, which is using audio for data analysis. So nothing to do with music, it's audio for data analysis. So I thought, if I can do sonification of coronavirus sequence and turn the coronavirus genome into audio patterns, can I then later on take the audio patterns and use it to make music? And that way I bring together the things I love, which is molecular biology and music, through this process of sonification.
So could we just take a quick detour into the science and the medical application potentially of being able to listen to a genetic code? Because that's a big part of your biologist job, isn't it?
It is. So I managed to publish my work in 2 science journals. And the idea is, rather than using visuals to analyze a sequence and to use colors and make things bold and to, annotate sequences with information, I thought, well, let's use audio to tell us where we are in the sequence. So I'm thinking of the sequence and I'm imagining I'm a protein binding to my sequence. And then the protein moves along the sequence and it reads the sequence as it moves along, in the same way that you read paragraphs in a book. Your eyes are doing the reading. So the protein's doing the reading. And when it reads the sequence, some sequences are more important than others. So we have sequences called start sequences, and we have sequences called stop sequences. And they indicate the start of a gene and the end of a gene. So I thought, well, why don't I use the start to start the audio, to start making noise? And I'll use the stop codons to stop making noise. And then the other thing is that you can read the sequence in 3 different ways. So you have one sequence, but you have 3 ways of reading the language. So I create 3 layers of audio, and I pick up stops and starts in those 3 reading frames. So sometimes you have 1 note, 2 notes, 3 notes playing, and you get this mixture of notes. And then I just started to map that to things that sound like musical notes. So I sonified it into audio, but I made musical noises. And then that starts that track, that journey into music. Does that kind of make sense?
That makes a lot of sense.
I think the more you drink during this talk, the more sense the talk will make. But I encourage you all to drink more, okay?
So once you've started building up the sounds, what do you do with it to take it into the music sphere?
Yeah, so within the science lab, I've generated this audio which sounds a little like music, but it doesn't have musical characteristics. There's no introduction, there's no middle 8, there's no build-up, there's no sort of a crescendo, it's just do-do-do-do-do-do-do. So I then take that audio into a studio with a bunch of musicians, so guitarists, bass players, synthesizer players, and we play it and we respond to the music you were hearing earlier with real instruments in kind of real time, and we start playing to it. And it changes the way we play, and it inspires us to make different sorts of music. Like, you've got this backing track that goes for 2 hours. So we could effectively jam for 2 hours, which would be totally indulgent. We decided not to do that, to actually try and give it introductions and middle acts and endings based on the sequences. And sometimes we play for an entire gene. So I think one of the genes lasts for 8 minutes or 6 minutes. So we did a 6-minute track from the start codon to the stop codon. And that was really fun, doing things like that. And the musicians loved it. They thought it was a really great way to engage with science. It was a great way to respond to the sequence. In these sort of terrible times, we actually found ourselves engaged and thinking about the music and trying to, in some way, I don't want to remove it from the science because I'm a scientist, but I actually want to use it to respond to the virus, not just in an intellectual way, but in an emotional way as well. And the cliche is trying to make something beautiful out of something that's so awful, you know. And I feel a bit guilty doing that. You know, I, as a scientist, there's no other way I can respond. I can't implement what I do towards a cure. I mean, I'm not that kind of scientist, you know. So to me, it was the logical response.
Fabulous.
And we have the same thing in terms of how do we respond, you know, even as a science hub, but also to generate fun, because we need to, you know, be able to celebrate life at this stage as well. So it's an interesting catch-22. We've got to look at the serious side as well as, you know, how do you get down and have some fun?
So a bit of a stray question. Apart from your own music, do you have a favourite dance track?
Oh, well, I'm probably dating myself. I quite like New Order and some of the Blue Monday, I love that kind of stuff. I don't dance to it in public, but I dance to it in my mind.
I dance to it in public.
And if I was to be brutally honest with you, I quite liked some early Donna Summer in the late 70s. I thought some of that music was pretty good, with that Morodo sequences in there as well, that really dance grooves in there. And Delight. I thought Delight were particularly good. That Groove is in the Heart.
Yeah.
A particularly good song.
A big fave.
Yeah. But I also like punk and prog.
Well, I'm afraid we're the same generation, so they all sound mighty fine. You know, we're all Brisbane people, so, you know, I always love to end something with the Saints with Know Your Product.
Absolutely. Know Your Product.
No, that's right. Know Your Product. That's right. No, My Product was actually an exhibition up there. Cool. But I always seem to throw up my hip these days when I dance to that one quite vigorously.
That's right.
Well, thank you very much. Thanks for being part of this, trying to spread some joy during these hard times.
No, thanks for inviting me to the ball. I'm pleased to have been here virtually, and I'm desperately upset that I'm not there personally.
Another time.
Yes, indeed.
Thank you.
Thank you.
All right.